Hello, my name is Cameron Ross and I'm a NexGeneGirls science intern. My research was at the Dr. Lauren Weiss lab at the University of California, San Francisco, where I studied the effect of shRNA knockdown of 16p11.2 copy number variant genes on the development of iPSC-derived neurons. 16p11.2 copy number variant, or CNV, is a deletion or duplication of a region on the 16th chromosome. The 16p11.2 CNV holds 29 genes that are previously known to be associated with various neurodevelopmental disorders such as autism spectrum disorders, developmental delay, schizophrenia, and epilepsy. There are several phenotypes that are known to be associated with deletions and duplications of this region. For example, macrocephaly seen in deletion carriers and microcephaly seen in duplication carriers. Our goal is to study which specific genes in the 16p11.2 CNV are responsible for these cellular phenotypes in carrier-derived neurons. The two specific genes that I'm looking at in my experiment are PGR1 and KCTD13. My first step was to perform an shRNA knockdown to reduce the expression of these genes in order to resemble the phenotypes seen in deletion neurons. We also had to introduce pyrimycin in this experiment to ensure that the shRNA would infect all of the cells. After this experiment, these images show the results. As you can see, the 16p deletion neuron shows increased soma area in comparison to the control, as well as the duplication neuron showing reduced neuronal size in comparison to the control. I used a software called Fiji Image J to measure the soma area and dendrite lengths of these neurons. As you can see in these graphs, the soma area for the 16p duplication of the KCTD13 gene was statistically significantly larger in comparison to the controls. However, PGR1 unfortunately did not show any statistically significant difference in size in comparison to the controls. After performing an immunostaining assay, these images show the protein expressions for the NPCs and neurons. For the NPCs, we used two proteins, nestin and PAC6, and for the neurons, we used two other proteins, TAJ1 and MAP2. These images show the separate channels of the protein expression, as well as the merged image of all of the proteins expressed in the cells. Again, I used a Fiji Image J software to count the cells and count the expression of the protein in all of the cells. These two images show the proportions of nestin and PAC6 in the NPCs. Overall, I found that KCTD13 does have an impact on neuronal morphology and rescues phenotypes in duplication lines. PGR1 appears to have no effect on neuronal morphology in duplication lines. The pyrimycin treatment also appears to have no effect on deletion and duplication-derived NPCs. In the future, more experiments should be performed to analyze the effect of pyrimycin on controlled lines. We will also need to perform more shRNA knockdowns without pyrimycin in order to analyze these genes' effect on neuronal morphology. Lastly, I would like to acknowledge Dr. Lauren Weiss for allowing me to research in her lab, as well as Dr. Aditi Deshpande for her mentorship. I would also like to acknowledge the NexGeneGirls for giving me this amazing opportunity.